Run. Fantastic. Good morning. Welcome to Neuroccino, everyone. Today we have one paper and one book review. There's more people dropping in. Fantastic. The way it works is you present a paper or a book that got you excited. You briefly tell us why you were excited about it, what it is all about, and then we have a chat about the paper or the entire book for that matter. Right, without further ado, Leah, do you want to go first with the paper? And for everyone watching over on YouTube, I will put the link in the chat on YouTube as well. Perfect. Thank you.
Okay. So this is a review called Where the Genome Meets the Connectome, Understanding How Genes Shape Human Brain Connectivity by, this is difficult to read, by Aurina Arnatkeviciute, say the name. And so it is about genetics and neuroimaging studies and where these two experimental approaches meet and how, and exploring all the, is a preprint, but I found it really interesting and really comprehensive for the field that I'm starting to explore a little bit. In particular, the review is divided in three parts. So we have the first part where they analyze the different twin studies, where of course you have a large part of the genome shared between the twins that are homozygote, and you can investigate the contribution of the genomes in phenotypes like neuroimaging measures. And then in the second part, they show how specific DNA variants, like little variations in different chromosomes, but they are recurrent, can correlate to some phenotypic variations. And so this is the association between the two. And then finally, in the third part, they analyze the gene expression and how this is pictured in the brain through transcriptomal atlas imaging and analysis. And it's really nice because in the correlation with the neuroimaging studies, they evaluate both structural imaging, like also the size of the brain, diffusion MRI, fMRI. So it's really interesting, and there are a lot of studies there that I didn't know, and here you can really see. So one of the main features is the hereditability of some phenotypes, and so the correlations with the genomics. And so in the beginning, you have this introduction about the hereditability and how also you can define that. In particular, there is this formula. This is like A, that is the contribution of the genetic contribution divided, so this is the proportion of the variation that you can, the phenotypic variance that you can explain related to genes, normalized with also the environment contribution and also the specific environment contribution to the individual. So this is a way of express. There is this age square index that expresses the variability, the hereditability, and so the variance of the phenotypes related to the genes. I hope it was clear. And why the environment also is important, because there are studies that show that, for example, age hereditability depends also on nutrition conditions, or they can change over time, and so really depend on the context. So it's not just genes, but also the expression related to the environment, and so this is important to take into account. And so a way to analyze this is consider the twins, and in particular the homozygote ones, and you have the heterozygote that share just half of the genomes, or also comparing to the siblings that are non-twins, and you have all this data in the you can find this data in the Human Connectome project. So here, as we were saying, there is the equation, this hereditability equation, when you have just a proportion related to the genes and the variance that put together all these factors. And so in the homozygote twins, you have that the genetics is shared in the whole. Otherwise, it's half if they are heterozygote. And here you can see the correlation. So in the homozygote one, you can really see that there is an agreement between the two twins. And so the genetics really play a role in that. And also you can investigate this population based on fMRI and the diffusion. Here I'm pointing to diffusion because they analyze the correlations with the genetics with the first peak of the functional orientation distribution relating to the fibers orientation, and also FA. And you can see how in the white matter the agreement is really, the readability is really high, so close to 1. Also really interesting, you can find this genetics factor also in the graph description of structural connectivity. Here you have in the x-axis the degree of nodes connectivity, K, and the different threshold that define the apes. And if you divided the nodes in the connectome in a reach, so there are the apes, the feeder, so the ones that are connected to the apes nodes, and the peripheral one. You can really see that in here's the distribution. You can really see that the age square index, so there is the readability, it's really high when you are an ape. And there is so an agreement between these the genetic contribution mainly in the most connected nodes in the brain. And also here there is the same approach, but based on fMRI data and different networks. And you can see that the range of readability is slightly lower, but is still present. And yes, as I was anticipating in the beginning, you have other important features that you can see that can describe the heritability. So the total brain volume, the cortical thickness, also the folding of the cortical layers, and the change over time, the fractional of FA, fractional anisotropy, and also there is also the analysis based on the overall in all the brain. So whole brain measures of diffusion. There seems to be like a better biomarker for the genetics than rather than specific tracks. And yes, here what I was saying. So it's in local white matter is the contribution, the agreement is lower than compared to global measures. And maybe going a little bit in, yes, also these there are also a lot of graph measures that like path length or clustering coefficients that have been investigated in these genetics contribution, and also fMRI, not just functional connectivity, not just through fMRI, but also MAG, AG studies, and but the agreement sometimes, so there are mixed results. They say sometimes they find an agreement with genetics, sometimes no, and here, as you can see, the range is a little bit lower than the ones with diffusion MRI. and this can be explained to the different processing pipelines or yes, just these results. So the structural connectivity seems to be a more consistent biomarker, let's say, for this analysis. And in particular, it seems that also in the different processing of diffusion MRI, deterministic or probabilistic, it seems that the probabilistic is slightly better in this, showing a higher repeatability in this analysis base concerning the graph measures. Then the second part of the review. So as I was saying, like the agreement between the genetics variations and phenotypes, for example, so using neuroimaging measures. In particular, they're called genome-wide association studies. And for this, you can use large data sets that put together these two features, like the Enigma project or the UK Biobank. And an acronym that is really used is this SNP, the single nucleotide polymorphosis. So here, maybe the figures are a little bit not really big, but I can try. So here you have the chromosomes and all the different variations that you can have there. And this is the correlation. What was that? The correlation between the different phenotypes, and in particular, then you can see like in each chromosome and in the different alleles, the two parts of the chromosome, if there is like a variation of just one nucleotide, for example A or instead of G. And these really local variations can be like more recurrent in people. And here you can see that they are more present in cases are the patients rather than controls, so seems to be important in pathologies. And you can correlate these with imaging measures like diffusion MRI and function MRI. And here it seems that they measured that more than a half of the significant correlations were related to diffusion MRI. And also you can see that the scale of the repeatability is higher in the diffusion MRI ones. In particular, here you see the one variation of this particular spot, this was the SNP variation, and this is the probability, the correlation with the right inferior longitudinal fasciculus, and how these correlate then in the whole white matter. And maybe I'm saying a lot of things because it's really compressed. So there are a lot of things to say. Maybe I can go a little bit faster. And then in the last part, so the transcriptomic analysis, these measures are a little bit more difficult to have. Sometimes are like postmortem, so you evaluate the gene expressions, not just the variations in the gene structures. And famous are really the Allen mouse brain atlas or microarray measures, also the Allen human brain atlas. And here, for example, you can see all the transcriptomic points related to the Allen human brain atlas, and then the partition of the brain and the expression of different genes. So here the idea is to create this matrix. So when it is genes against genes, so you have the correlations of the gene expression in the different regions of the brain. So you have like you can have in a structural fMRI or functional structural association matrix or fMRI. You have this matrix in this case the when you have the different regions, you have the correlation of the different genes, and then you can compare the gene matrix with other measures like like the structural matrix ones. And here again, you can see these the different behaviors related to the importance of the nodes in the graph. So the apes plays again an important role in this correlation. And also here you can also see, it's really explained in a good way in the text, but now I'm gonna like end up, otherwise there are too many information. So there are informations, these are the examples are related to the Turner syndrome when you have like the X chromosome loss, or also depression, how the different genes express in the different brain areas, and in schizophrenia. Here there is the correlation of the gene expression and diffusion MRI, the connectivity measures. And so yes, this was the general idea behind the review. I don't want to spend, maybe I have taken, I took for a long time, but yeah, this is the idea.
Thank you, Leah. That's a very rich paper.
Yeah, I'm trying to synthesize, but yeah, a lot.
I could tell, not an easy job. Great. Before I give the questions to the floor, I can see Valentina has a question and Michelle is applauding. I just wanted to highlight that for those watching us over on YouTube, feel free to write your questions in the chat box and I'll bring it over here to Zoom. Right, questions. Valentina, go ahead. Oh, I think she is frozen.
Okay, now maybe no.
Are you with us? Yes, no, maybe. Yeah, you.
Yeah.
There we go.
Yeah, sorry, it froze. I missed your introduction part to my question. I'm sorry.
No worries.
Yes, very, very interesting paper. Thank you, Leah, for presenting it. I wanted to ask you, so can one predict the cognitive development in the cases of twins by watching at the genes expressions, or in general the cognitive functionings of an individual by decoding the genes expression?
So when you talk about development, maybe you are looking to longitudinal analysis, or okay. Here there is it's just the test retest in the twins. So I don't know if there are longitudinal analyses in that. Here in the cortical foldings,
there is no longitudinal, no, in gene expression. So
so if they change over you mean like that the genes can change some variation that can occur? No, I don't think they can occur over the time. Even the expression, maybe they can change.
There is
maybe I don't know. Hervé, maybe you.
Yeah. So for the gene expression, the ALEN Atlas has a developmental brain expression maps. So you can look at different regions and the expression of certain genes based on age. So you can look at the curves and look at the it's not longitudinal, but you can see the developmental part of the gene expression.
But that is in the expression. It's not that like in the genes some variations, some alterations can like develop in time. That is fixed, no?
What do you mean by fixed?
So things like, for example, if I look to if I process the genomes like and I have like all the nucleotide sequence, then if I do that after 20 years, that is fixed, or we can have variation of that, not fixed.
For the variation, I'm not sure.
Indeed.
Yeah.
No, I was really talking about the cognitive development, not the gene expression development. So by looking at the gene expression of a child or two twins, you can predict the cognitive development of the individuals.
Ah, so like cognitive measures.
Yeah, the problem is to get the gene expression of the brain on a live person. So that is complicated. So what you can do is to get the gene expression in the blood, but it's a proxy of the gene expression of the brain. So it can be not the same. So the problem of that, the expression is based on the tissue you are looking at. So it's kind of complicated sometimes to get the right correlation.
Indeed, like when they evaluated the whole gene correlations, the document, they looked at the single nucleotide variation related to different phenotypes. So maybe they did that. There are studies also on autism, and I can see the Michelle paper ready.
You just sneaked in there. Yes, go for it, Michelle.
Hey, you gave like the screen. I was getting ready.
I just wanted to show the people whilst we're having a chit chat rather than paper.
We're on a tight schedule.
That's fine. We're moving on. We're moving on.
Well, I had a question. So they're really promoting white matter as being more linked with genetic expression, but I was wondering whether this relationship can be driven by the fact that diffusion imaging, or the way we assess connection with diffusion imaging, is less noisy than standard fMRI, cortical thickness measurement, and any other or functional connectivity. What do you think? How this could have had impacted these results?
So here in the paper, they discuss more the biological meaning, correlation. So how how genes really can shape the connection. They use there was a sentence, but yeah, I can find that is like really also in the myelin production. So they explain this in a biological way. But yes, also in fMRI, I guess also there was a paragraph where you can see that these mixed results based on fMRI are related also to the different pipelines and probably to the noise. But also like a simple measure like the size of the brain is shown to be like significant in these gene correlations. So maybe yes, can be either that is a more direct measure, so there's less manipulation, so you can infer in a more direct way the correlation with the genes, or there is maybe like you can explain this in a biological, let's say, way. So it's more in the structure than in the function, in the functioning, indeed, like in the transcriptome correlations, then you find the correlation with fMRI. So it can either be the noise present in the data, or more that maybe the infrastructure, the structure is more related to genes, and then the functioning more related to the expression, the different expressions of the genes. Indeed, the example that they show are based on schizophrenia or depression, where you look to the different gene expressions. So I think there could be like both the reason why you have this difference. To me, it makes sense also from biological, let's say, point of view.
Thank you. I just saw in the chat that I stole the question of Hervé. Do you want to add something, Hervé?
Oh, yeah, that was yeah, because they made a lot of distinction between structural and functional heritability, and so I wanted to know if they had more explanations about it. And do they give recommendations for, because they say functional connectivity is not heritable, do they give some recommendation how to use functional connectivity to make association with genetic and
So they say that the results are mixed, not that there is not hereditary. And one pipeline that they show is this one that when you have this matrix with the gene expression and you compare the gene the correlations between the different regions in the gene expression with the correlation of the different regions based on fMRI. So this is the way to compare it using the same framework. So the brain parcellation based on the human Allen atlas, and then you can compare the correlations between the two, because one of the problem is that there is a lot of self correlation. So like and if you want to compare the data set that there is like a lot of past, like a lot of correlations between it with another measure that presents like more correlations in itself. So this is the tricky part. They can also bring to false positive correlations, but they say that now with also big data sets, you can assess the significant correlations. But yes, they don't say like you have to do this. They show like how it has been done in the literature.
Okay, thanks.
Any other questions? No. Moving on to the next book then, How the Brain Makes Decisions. Michel.
Hello. How the brain makes decision. I'd like to know. And this book by Thomas Boraud, which happened to be the director of our institute, no bias here. This book is great because I'm not coming from this training of people working on decision making, and it's a good introduction because it really starts from scratch, from the original equations from. Can I move the page? There you go. With the original equation from Bernoulli, where according to a reward, you will progressively make the decision to move, and this is a matter of probability, and because you expect this reward, you have a higher probability to move, and how this can be modeled according to whether you're gonna have some gains or you're gonna have some losses, which are either money or positive or negative feedback. And by doing this model, you can try to see whether there is a reference point that crosses the zero midline, which in theory should, but practically you have numerous experiments that are like discussed inside these books that are showing that we're not so rational about our decisions, and if you model the way we decide things according to gain and losses, not everybody is centered on zero, and this is really representative of a fundamental trait of your personality. The book is quite well organized, and I lost my notes. It's an interesting story, and you can see how the main author is really battling and fighting inside this book between working his entire life on defining the biology behind decision making, and on the other hand, having results that really clearly shows that not everything is explainable with biology, and there is a part of stochastic noise in decision that might be attributed to free will. And that's probably like the inherent like fascinating part when you read the entire book, is like how you can see how from really a solid biological investigation and entire career exploring this, it comes down to the conclusion that most of our decision can be modeled, but a part of what is going on, and it's like you get all these kind of gimmicks that are funny, is related to either randomness or true free will. So that's about the theories, and practically in terms of anatomy, this is probably the best book I read in terms of schematics and drawings about the organization of the interaction between the cortex and subcortical nuclei, in terms of the way they actually dynamically exchange via a loop of connection and excitations and inhibitions. So you have, every time is adding another layer of complexity into the process of decision making, and every time is adding like more structure into this kind of schematic. And the truth is, decision making is really based on, if we want to be rational, on two things, which is like rewards. It's like, are you going to gain something by doing it, or are you going to lose more or less by doing something? And the other part is learning. You make your decision based on what you learn. And sometimes you don't even think about it, but the decision that you made is through simple associations. Typically you get if you had to choose a tool and if you had to choose a color, most of you went for a red hammer, and that's just because it's it's like the first occurrence. And some other people have a different idea, and they sometimes have a different culture, or like say controversial, or they wire differently in the brain. Another interesting part that I found is like really pushing the discussion quite far. He also had inside the decision making this other, so there is an evolutionary part. He also had inside the decision making another dimension, which is the mirror neurons, and the way you make your decision is sometimes the way also you can project yourself in the boots of somebody else. And they have a fantastic experiment for that. So I just want you to try that. And these directly test how much you take into account others and mirror neurons into your decision making. So the test is going this way. You're going to have to give a number between 1 and 100, okay? And the goal here is for the number you give being the closest possible to half of the average of the number of everybody else. Now I'm going to do it again. You give a number between 1 and 100, and the number that you have to give has to be the closest possible to half of the average of the numbers of the others. So you can write this number in our chat. On YouTube, you can write it into 2, 22, okay, 10, 25. Anybody else? There is only one right answer. Okay, 12. Okay, that's pretty good. It's pretty good. So basically, this is an infinite loop, and so the smaller the number you give, the more you reflect into the mirror neurons and exchange with the others. So if you put 25, you think that in average everybody is going to give a number between 1 and 100. So the average of everybody is 50. If you want to provide half of that, you're going to say 25. And this is good thinking of it. But the problem is everybody is thinking the same way in principle. So everybody should say 25. And that's thinking about the other, already thinking that they're going to give half of what the other is going to give. So if you said 12 or 12.5, you are first, well, second level of mirror neurons. You think that the other thought about it. Well, it's the first level of mirror neuron because you think the other one had mirror neurons, so they give 25. So you give like the alpha of this 25. But if you give half of that again, it's because you think that the people thought that in average, everybody will think that alpha will be 50, but so they gave 25. But they think that everybody will think that when give 12, so you give 6, and you had your second or third level of mirror neurons. And that's an infinite loop that bring you to the ultimate answer, which is 0. 0 is not between 1 and 100, so yeah, that's correct. And the answer is actually below 1. But you can also say 1 if you want. And so you have all this model of reinforcement, and it's really, it's a nice story. But then you also have like those nice like discussion about, is really how far are we going into science being rational and forcing people to be rational about everything, or can we believe into things not necessarily always being rational? But if you want to explain things, you got to explain it from the rational way, because if you don't use a rational way, there's nothing to explain. It's just the way it is. And so in this discussion of the rationality is also relating the decision making with different pathology toward the end of the book, where he's going to discuss like obsessive compulsive disorders, Tourette syndrome, and he takes again all these nice graphics of loops and interaction between brain regions, where he's showing what is known to be dysfunctional in these different neurological disease and how it leads to different syndromes. And you get, of course, Parkinson's disease, which is one of the most frequent disorder. And you get the hyperdopaminergic syndrome, which would be as a reverse of Parkinson in principle in terms of neurotransmitter. The book ends up very nicely on free will, and it is really a progressive discussion that is bringing all along the books that I found really interesting and made me believe that we cannot explain everything. And yeah, that's about it. Toward natural history of irrationality. That's it. If you get questions, be happy to answer.
Lovely, thank you. Any questions?
Yeah, I have one. It's about the free will, actually. I would be really interested to know a bit more about what he argues, because it's a very complicated topic, and usually it depends a lot on the definition you give of what is free will. And so
you're absolutely right. So what is going on is like so he's been working in animals for a long time, and you can have the best model, really the best model you can have into determining the relationship between the neurons, the decisions, the reward, and everything in your animal. It works most of the time, but for some reasons, and he says that's why it's stochastic, sometimes the animal's going to choose the other way, and there is no way to explain that, except that is there like random bugs in the brain? You can say that, but it's not explaining it, saying that, oh yeah, you just hit the button of the random bugs, and surprisingly this time the rat went the right side instead of the left side for no reason except that there is a random bugs in the brain. These random bugs it's a free will.
Nah, I'm not convinced, because to me, I can easily imagine that. Well, imagine. I think it's kind of the way the brain works. The brain is always in interaction between the inner thought, let's say, and the sensitive inputs, and there is always an interaction that creates random fluctuations depending on what the animal feels and what he feels internally, and what he sees, and what it wants. And it's a complex interaction that I think is very hard to model with the models that we have. So we can have something that is fairly accurate with the broad, okay, the broad understanding of, okay, what does he want usually? But there is the whole interaction of how does it feels at the specific moment, and how does it interacts, and what it remembers, that will play a big role into that also. And that is very hard to model. And I think that saying it's just randomness or free will is a bit reductive. It's just a very complex system.
It's a belief then. You have the belief that we can model that with a better equation. Thomas Boraud is discussing the fact that actually it doesn't matter how good the equation is, there is a part that is not rationally explainable. That is defined as free will. But in both cases, you know, that is infalsifiable, infalsifiable. Yeah. Just because we'll never get that perfect equation ever. And we'll never be able to prove from a fact that, you know, this noise is actually truly free will. This is as part of the spirituality. So it's a paradox where both answers are right. It's just a matter of belief.
I kind of hope that at some point we can have at least a simplified model that shows this kind of behavior, and then we can say, okay, we can maybe not explain it on very complex brain like mammal's brain or stuff like that, but in simpler brains with fewer neurons, if we see the same kind of randomness appearing, but yet we can find the origin of it, and we would always extrapolate and say, okay, we have the same kind of behavior here that we see here. Here it's simpler, so we can explain it. It's probably the same here. And I think we will come to that at some point. I mean, I hope. It's still something that has to be done, but I'm fairly confident that with the advances in a few decades, we might get there.
Let's have a beer in a few decades.
Oh yeah, we'll do that.
Solid plan.
Talking, I was thinking that maybe the randomness or the free will is not based on the reward, like on the future. You can model that to look into the future. So what is affecting you if you make that specific decision, but maybe it's based on the past. So maybe it's based like the decisions that can be like different between each individuals are based on their experience. So if in the past learned that specific thing can like hurt you, you don't want to go there. But maybe another individual didn't have that experience. So maybe it's not just the future. It's also the past, how you experience that can frame your decision. That can make the difference between the individuals, because not everyone had the same past. So you can see the same future, but not the same. No, makes sense.
It does make sense, and I'm sorry, I want to maybe a little fast on this, but so like the chart and the equations that you have in the book progressively increase their complexity, starting from reward and heading to that learning, which is the past experience you're talking about through avian and non-avian learning. And then is expanding this to mirror neurons with like the fun experiment that we did together that can also influence the way and the outcome of what you're going to decide. And those are essentially the three main things that can influence your decision. And if we head to that, the way people feel, maybe as Victor has been mentioning, that's also a good point. Maybe we can explain 99% of the decision making in the simpler animals. But the argument that Thomas is doing in his book that I found like extremely surprising from somebody who spent his life on this is that for surprising reasons that there's still a part of variance that remain unexplained that is open for discussion for is it free will or the model not being good enough. And I am under the impression in his books that he's going toward free will, but I don't know.
Does he discuss in the book the role of inter-individual variability on the anatomical level and or the decision-making level?
Yes, and I think as a matter of fact, I sent you a little section of his book. I don't remember exactly what he said because it took me a month to read, maybe a month and a half to read this book because I'm trying to do it in between like parenting. Yeah. He's speaking about he's covering the entire field really. You have inter-individual variability, you have like the story of free will, you have like learning, you have reward, you have mirror neurons. It is a pretty good book from the perspective of the decision making people that are really want to explain everything from the decision making. So it's really a good window of understanding, and it's actually fairly understandable for people that are not expert like me in the field, and like many chapters are really easy to read for people that are not even expert in the brain. So it's very comprehensive. Any other questions? Cool.
Right, in that case, let's call it a day here. Just a quick reminder, we have a CNS talk this week on Thursday at 4 p.m. by Alfredo Spagna from Columbia University about imagery, visual mental imagery. So do join us for that one. Everyone is welcome. This is this week, Thursday at 4 p.m. And thank you very much for coming today and have an exciting week in science. See you next week.
Thank you. See you.